We got a pretty tight agenda today, and there's a lot to, 3 talks again, and there's a lot to cover, so I don't want to spend too much time with an intro, but let me get started. My name's Tim Nedwood. I work for ExxonMobil, and I'm going to be kind of the host here today. This is the Dispersants Herder webinar series. This is webinar number 2 out of 10. I'll just give a brief kind of intro on why we're doing this. I mentioned it last week, but we're bringing together kind of as broad a group of people as we can. So we have academics who are interested in oil spill response related research. We have industry folks, subject matter experts from industry, from the consulting world, and we have folks that work for government. And the goal of this thing is to bring everybody together so we can talk about research that's going on and get different viewpoints on some of the research that's going on. This week, we have 3 talks. We're going to kind of expand on some of the topics that I brought up last week on testing protocols. So the first talk will be by Roger Prince, and I'll introduce him in a minute. But he's going to talk about biodegradation of oil. The next talk will be by Tom Parkerton, and he's going to talk about oil and dispersant toxicity testing. And then the final talk is a talk by Benny Gallaway, who's a fisheries expert, but he's going to describe a technique that he's developed to bring toxicity data into a way to predict population level effects to a fishery. So those will be the talks. We're going to hold, the format here is like last week. We're going to have each speaker will have about 20 minutes to talk, and then we'll have 5 or 10 minutes of question and answers. So if you see the question and answer button on your screen, that's the way to send questions to me, and I will ask questions as they come up. I'll get through as many questions as we can before we have to go on to the next speaker. But I'm going to be logging all the questions, and I'll send the questions to all the speakers, and we'll try to get answers to each question that wasn't answered, and maybe some more clarification on some of the questions that were. And I'll circulate those questions and answers to the participants sometime maybe later this week. So with that, let's get started. Our first speaker is Roger Prince. And I'll give you a brief introduction here. Dr. Roger Prince was a senior research associate with ExxonMobil Biomedical Sciences in New Jersey until 2016. He was Exxon's lead scientist in the monitoring of the successful bioremediation of the Exxon Valdez spill, and also did field work on experimental spills in the Arctic. Prior to joining ExxonMobil, he was a visiting faculty member at the University of California in Berkeley and on the faculty of the University of Pennsylvania. He's published more than 370 papers and chapters and referee literature, which is an amazing thing for somebody in industry, I have to say, and was awarded Stanford's Farrell W. Lytle Prize for contributions in synchrotron spectroscopy in 2000, the North Jersey ACS North Jersey Chapter's Lifetime Achievement Award in 2007, and the Waxman Honorary Lectureship of the Theobald Smith Society in 2013. So with that, I'll turn it over to Roger. Okay, Roger, you're on mute. There you go.
There we go. I think we're in good shape.
Yep. All right. Screenshot.
And I'm going to share the screen. It's not full screen yet, correct?
No.
So I'd like to talk to you about oil biodegradation in the context of oil spills, right? There are lots of other contexts you might study it in. I'm going to talk about oil spill response. Biodegradation is by far the dominant process removing oil from the environment. It's not really surprising, right? The average age of crude oil is about 100 million years, some older, some younger, and it's been seeping forever. And so bugs have taken advantage of this very rich source of energy. So there's been a strong selection pressure for organisms to exploit hydrocarbons, and we now have several hundred named genera of bacteria, archaea, and fungi that can grow on them. They're found everywhere. There's aerobes and anaerobes. We're mainly interested in aerobes for oil spill response. But oil is a very unusual food, and this is a deep truism that, alas, many people forget or are unaware of. It's a very rich source of carbon and energy, but none of the other essential elements for life. Right? As we all know, you are more than carbon and carbon. You've got nitrogen, phosphorus, iron, a whole litany of elements are essential for growth. Now, fortunately, these are all in the sea at low concentrations. So if the oil is at low concentrations, there's enough of everything and the microbes are content and consume the oil. But oil on the shoreline likely requires added fertilizers because there's so much oil. It's a whole different ball game. That was what happened in the Valdez spill, but we won't talk about that very much today. And the other thing is oil is essentially insoluble. Only the small alkanes, less than about C6 hexane, and 1 and 2 ring aromatics have significant solubility. And it's easy to see this is true, right? Beached oil that's been stuck on a shoreline for years, when analyzed, can often contain its full complement of, say, phenanthrenes, 3 ring aromatics, even though they have a finite solubility. And there's clearly an issue that we aren't aware of or don't have much control over, which is the solubilities reported are at equilibrium. And they are indeed, like most thermodynamics, they tell you nothing about kinetics. And it's clear that the dissolution, despite the fact how big the ocean is, dissolution from oil is very slow. So that means that biodegradation mainly takes place at the oil-water interface. And that, of course, means that that has to be as big as possible if you want the oil to be degraded as quickly as possible. Oils are very complex. As a rule of thumb, though, it's fair to say they're usually around 30% linear and branched saturates. They're about 30% cyclic saturates and about 30% aromatics. And the remaining 10% are polars, of which about half are typically what we call asphaltenes, really big, very dark molecules. It's important to recognize that when it's called an aromatic, that means it has at least 1 aromatic ring. It may well have all sorts of cyclic rings, all sorts of alkyl foliage all over it. But the way we separate these things, that would go into the aromatic bucket. Similarly, the cyclic saturates, they have tremendous alkyl substitutions, but we separate them this way into cyclics. Only the linear and the branch saturates are guaranteed ring-free. How do you analyze all those things? Well, there's lots of different techniques. The key one is gas chromatography. And this on the right shows 2D gas chromatography, which is jolly clever stuff. And all I want to show there is that you can discriminate the oil, divide it up into a whole bunch of different buckets. And we know the sorts of structures that are in those buckets. The key thing that I want you to go away with is that when you let bugs loose on that at the right concentration, a few ppm, the microbes consume essentially all the oil components except the hopanes. Those 2 circles there are components of Corexit. This is an experiment that had Corexit in it, but they also have been consumed by the end of the experiment. It's important to remember this is not looking at molecules bigger than about 40 carbons, and it's not including the asphaltenes and any other resins, things that don't enter the GC column. But of the hydrocarbons we can measure this way, it's not unusual to see more than 80% degradation in a couple of months. So the next question we're going to ask is, before we make much more progress, we have to really consider why are we doing the experiment? If we're doing it to understand a metabolic pathway, that's a whole different ballgame, right? If you want to understand toxicity, you need well-characterized organisms. But if you want to help oil spill response in the context of biodegradation, the real issue is they need to be relevant to the real world. And there are several points here that need to be borne in mind. Remember that different nutrients and concentrations select for different microbes. I mean, this is well known for higher plants, right? If you go to your local garden store, you'll find different level mixtures of fertilizers for different plants. Your lawn fertilizer is almost pure nitrogen with a pinch of phosphorus and potassium. Whereas ones for getting your tulips to bloom nicely is rich in phosphorus and has almost no nitrogen. And that's because different organisms have different preferences, different needs, and you will skew the system depending on what you add. It's also important to remember that mechanisms of lethality often have nothing to do with effects of low concentrations. Right? Aspirin cures headache. 2 pills will cure your headache. Taking the whole bottle will kill you. And it's not because you've made your brain really, really pain-free. It's a totally different mechanism. Scotch whiskey, right? 1, 2, or 3 glasses will make you feel very euphoric. The whole bottle will kill you. And again, it's not from an excess of euphoria. It's death by a different mechanism. And in both of those cases, the difference in concentration is only of the order of 2 orders of magnitude, right? It's 2 aspirin versus 200. It's even less than that for alcohol, right? It's 2 glasses versus a bottle. Lawn fertilizer, right? If you were to put 5 times as much fertilizer on your lawn as is recommended, you will kill it. So there's a big issue here. That mechanism of death is not to do with growing too much, right? It's a different mechanism. So you need to be aware that the levels that are in the sea can't be mucked around too much without causing big effects. So now I'd like to just run through the different experimental issues if you're going to do an experiment. For us, by far the most relevant thing to do is to use seawater with its indigenous organisms. The way we do it is we collect water from the New Jersey shore. We collect it close to shore. We bring it back to the lab. We're so exhausted, we let it settle overnight with a gentle aeration with one of those aquarium stones, things that very gently bubbles, right? In the morning, whatever silt and sand was in suspension has settled out. If there were to be any big lumps of seaweed or even little fish, we wouldn't use them. But then we just use the water, we don't filter it. Using enrichment cultures or culture collection organisms is suspect at best if you want to understand the real world. I will point out that some papers even used terrestrial isolates and then said, oh my goodness, they don't like oil. Well, they love oil when it's terrestrial. They don't like it when it's in seawater because they don't like seawater. That's the issue. And if you're going to follow microbial succession, it's really important that you don't diddle with the nutrients. Otherwise, you will certainly see a different succession of organisms than if there were lower levels of nutrients. And as I say, try adding too much fertilizer to your lawn. It won't necessarily not be green, but it won't be grass anymore. Oil concentration. This is one that's most important. But it's also important to think about what sort of oil it is, what the composition of the oil is. Remember, if you're going to do a dispersant experiment, with the best will in the world, under most circumstances, certainly all ship spills, they won't be spraying dispersant until at least 12 hours, right? I mean, it's just silly. In that time, spilled oil will have changed substantially. Depending on how thick the slick is, it will have lost most of the compounds with smaller than 15 carbons. If it was coming up from great depths, like the Deepwater Horizon blowout, then the BTEX and the C1 to C6 alkanes will all have dissolved in the water by the time it gets to the surface. So there's no point in doing experiments that you think are relevant to the Deepwater Horizon, dispersing oil and dispersing it with BTEX in it. There isn't any BTEX in the oil that's being dispersed. The concentration is a really big deal, right? Dispersed oil dilutes to less than a ppm in hours. You've seen pictures and you'll see them again. And the thing that is to me the most sad, but I guess it shows how you can abuse analysts. The huge database of the Gomery thing, only 126 of 20,000 samples had enough oil that you could actually analyze. If I was the analytical guys, I would have gone on strike, right? I mean, they measured 19,000 plus samples with nothing in them. I would be very cross. There is a problem for experiments, and that is that GC analyses need 10 mg per mil in methylene chloride. Now, you only need 1 microliter of that for most GCs, but that means, so you don't need the whole mil, but you need that concentration. So you need to use large volumes. We routinely use 2 to 3 ppm in 4 liters. Even that's high oil, but it's the best we can do. These are far too low for gravimetric analyses, so we use GCMS with Hopane as a conserved marker. If you're going to do experiments about beached oil, you need to think about what the oil will be like, very evaporated, and what concentrations. Beach is difficult to think about, right? Because most of the beach is, of course, rock. It may be ground up rock with big surface area, but at the oil-water interface, the oil is very concentrated, and there's a lot of it. So it's a tricky thing to think about, but we've done effective experiments there. I'm very cross about WAFs and CWAFs because I think they mislead environmental thinking. I mean, they were done for a jolly good reason, right? Oil is not very toxic. And if you just use whole oil and try and do a tox test, some of the little critters die because they're physically oiled, which even toxicologists who are delighted to kill things don't think is really in their bailiwick. So they designed this system where the oil stays on the surface, they stir it with or without dispersant, and then they decant the oil through a tap at the bottom. As you'll hear from Tom later in the talk, in his talk. There are lots of issues, but a major one is that all the soluble things will dissolve from that oil, right? There's enough, almost certainly, well, you'll get up to saturated benzene in there, whereas hardly any chrysene will dissolve. But then there will be droplets, and they are honest about calling it accommodated fractions. And the real issue is how small must the droplet be to be accommodated? And the answer is, all depends how long you wait. Even the most dilute system of droplets eventually will come to the surface if it's sterile and the bugs don't have a go. It may take days. But oil always floats, right? Dispersants do nothing magic. They don't change the density of oil. The only reason oil stays submerged when it's dispersed is the droplets are so small that their buoyant density isn't enough to beat out turbulence. Nutrients, this is another one that is really a key issue. Seawater does provide those essential nutrients, but at very low levels. Even micromolar is generous. I'll show you some real data in a second. But the media that microbiologists have used for quite other reasons are staggeringly high in nutrients. Therefore, almost all of them would kill any passing fish, right? They're just nothing to do with the real environment. And I confess, I've done experiments with bushel hulls. I only slowly twigged what the problem was. And remember this issue about what you use, nitrate, ammonium, you're skewing the system. Some bugs prefer one, some another. You're not mimicking the real world if you add high levels or even any levels of nutrients. Here, for example, are the ocean nutrients from these enormous experiments that are done by oceanographers. And this is between 0 and 40 micromolar. And even then, you can see most of it is less than 10 micromolar nitrate. And then there is, in most places of the world, ammonia is at least tenfold less than that. But that's still fine for the bugs to degrade oil, as many labs have shown. That's enough nutrients to degrade the 1 ppm oil that happens when you allow dispersion. Temperature. I think it's safe to say that microbes are adapted to their surroundings and that the sea is somewhat mixed. So that means then that, and we have found this, we haven't studied it really thoroughly, but microbial populations are likely adapted to growth throughout the year, whereas individual organisms likely have different optima. But since we're working with populations, it turns out that oil degradation is not very temperature dependent, certainly not a Q10 type system. I would say that if you're really interested in polar or tropical regions, it would make sense to get your bugs from there. But for temperate regions, I would say that it's very unlikely they're very different in different parts of the world. Light. Light's clearly an issue because it can photopolymerize things. It's important to recognize that's what happens when roads, tarmac roads set up. The reason they don't let you drive on them for a couple of days is not because they're, quote, drying, they're actually photopolymerizing. Otherwise, the surface would not become robust and hard. And that's what happens with oil left for a long time. But all the experiments we're interested in, oil is dispersed within a day or two of release. After that, oil becomes less dispersible. And that's mainly because as it evaporates, the dispersant no longer can penetrate the oil when it's sprayed on it, it bounces off. So there's always an urge to do the dispersion as quickly as possible. And so photooxidation is probably not much of an issue for what we're talking about. Certainly, it will polymerize surfaces that are there for a while, like beaches. And there's no doubt that black oil on a rock gets ever so hot and becomes essentially pasteurized when the sun comes out on shorelines. But on the other hand, light is going to be important if you're interested in marine snow, because most people agree that most of the polymers in marine snow come from phototrophs, diatoms and algae. And so they obviously need light to grow. But that's a bit of a challenge in the lab, right? Sunlight is vastly stronger than lab lights. 1500 versus about 50 is the most people grow things in the lab. And the glass absorbs UVB. But on the other hand, that means, okay, you can only mimic what goes on at say 10 meters. But as long as you're aware of what the issue is, this is a tractable experimental variable. Dispersants. The first thing to say is that dispersants are actually used at an incredibly dilute level. right? I mean, they always talk about 1 to 20, which is just aspirational, wishful thinking, because the oil is never a homogeneous layer. But it is 5 gallons per acre, that number we have. And that's a teaspoon per square meter, right? It's nowhere near a coat of paint. It's way more dilute than that. So that means if you want to add dispersants in an experiment, it's best to add it to the oil in the lab before you do anything. Otherwise, these amounts are just so dilute, you can't really handle them in the lab. There's no point in adding dispersant more than once, no point in adding it to beaches, no point in adding it to the bottom of the sea, mesocosms mimicking that. But a key thing is that oil will disperse with mild stirring even without dispersants. So if you want to study the real effect of dispersants, which is to stimulate degradation, you need to do experiments in a floating boom, as I've shown there. Here's an example. As the floating slick kept inside that little boom, you get evaporation. You can see 14% lost in the 7 days because that oil wasn't fully weathered. We used one that was the sort of oil that would be dispersed, would be a target for dispersant. But if you disperse it, you can see it degrades really rapidly. Just a brief thing about dispersants. It's key to understand that the lab tests are excellent for what they're supposed to do, which is to distinguish between good and bad dispersants. They are not indicative of what happens in the real world. They are essential for registration. In the real world, at OMSIT, where we would perhaps be if it wasn't for the virus, this is a picture of the big tank at OMSIT. We routinely do experiments there. You can see in the bottom left of those photographs, that's oil floating and the waves are just coming along. The second picture shows dispersant has been sprayed on the top. And the first of those waves, they're not breaking great white horse rollers, right? They're just mildly breaking waves. And you can see that the oil has changed color to cafe au lait as the oil begins to be dispersed into the water and to scatter light. On the left, you can see a whole range of different oils. The key thing there is that the brown bars are the efficacy in the lab tests with the baffled flask test. And you can see that some of them are really poor. In the real world, even they work well enough. The green ones are onset data. The best test to mimic the real world is perhaps the XDET test, but that's not regulatory approved in many places. So that's just a useful thing to know if you want to mimic the real world. So finally, I would say that the most key thing to remember is that you've got to recognize that it's only useful for oil spill response if it's relevant to the real world. The big issue is to recognize the dilution that follows dispersion and not to add anything that wouldn't be added in oil spill response. Add dispersants if you like, that's perfectly fine. You know, that's what goes on in the real world. But people don't ever consider start my video. I haven't got a video. So this is a final bunch of things. I recommend real seawater, an appropriately weathered or biodegraded oil, a natural temperature, equivalent more or less to what it was that seawater was seeing before. Roller tables if you want to study marine snow. Even mild stirring disrupts marine snow completely. Light, no additional nutrients, few ppm oil, assays of biodegradation based on a conserved marker. Thank you very much.
Thanks, Roger. I turned your video off, so that's why you got that question, because I turned it back on at the end so we could see your long hair. But we have time. We have a time for some questions, and I have a couple here that I'll start with. One's a comment that the 12-hour delay for initial application time is true in most of the U.S., but not in Hawaii, where they have a requirement of 4 hours or less. So we may have relatively much fresher oil if there's a.
Oh, well, great. I mean, my comment would be that's exactly. Let's model that then. That's the experiment we need to do, right?
Yeah, certainly the faster is better. Speed is the key. And it's not just the weathering of oil when you let it sit out in the ocean, right? Mother Nature does things you don't want her to. Okay, here's another question. Roger, have you done actual experiments to discern the relative importance of photochemical reactions of spilled oil biodegradation?
No. I'm afraid we've done nothing much about that.
Yeah. Okay. Here's another question. Since biodegradation is a surface area dependent process, should droplet size be measured drew out biodegradation experiments? And what target droplet sizes make sense to you in an experiment?
Well, I would say we can't adjust. In the real world, we have no control over droplet size, except to say that we want a good dispersant at a dispersant to oil ratio that encourages the right size droplets. Right? That's where one of the good reasons for the 1:20, that tends to make droplets that stay dispersed for a long time, even without stirring. If you make a 1 liter bottle and put 2 ppm oil in it, even 50 ppm oil, and give it a really good shake, even without dispersants, most of the oil will stay as tiny droplets for a good long time. So the people who claim to have measured, as you know, the Norwegians have a device that makes droplets of a believed to be a known size. If you look carefully, you discover they lose half the droplets in the week or so of incubation they're doing. So it's not quite as clear cut. But we have no real control over droplets in the real world. So I would say in theory, the smaller the better. We like to believe they're a bit smaller than 70 microns, but we have no control over that.
Okay, another question. Would you please elaborate on the role of photooxidation? I might have missed your statements.
Photooxidation, the main thing it does is polymerize aromatics, right? It moves the aromatics from being aromatics into the asphaltene, the polar fraction of the oil. We don't know much about the biodegradation of asphaltenes or any of these polar molecules, mainly because we don't know what their structures are, and we don't know how to analyze them, except with these really high-end machines that give you 80,000 atomic compositions, but certainly no structural information. In general, it's polymerization of aromatics. That's been widely agreed. There is some indication there is secondary phenomena, but I would say that, of course, it starts as soon as any molecules see light, right? Photochemistry happens as soon as light begins, but there's not a significant buildup of these products in oil, in floating oil, over the first few days. They build up over quite a bit of time, and it does seem to require UV light, bright light. So for a floating slick, we don't think, and we have, I confess we haven't yet done experiments that way, we should do more, but we don't think the photochemistry has much of an influence at the beginning. Certainly on a shoreline, photochemistry is definitely your enemy. It's polymerizing the surface, making the rest of the oil unavailable to the microbes that otherwise would consume it. That's one of the reasons oil stays so relatively fresh when it's on a shoreline. It's typically encased in a polymerized shell of photooxidized products.
Okay, Roger, there's a couple more questions, but to give everybody a fair amount of time, we'll log those, as I said, and get them to you, and we can get answers, but I'm gonna move on. Tom, Roger, thank you. Thanks a lot, Roger, that was an excellent talk. Our next speaker is Tom Parkerton. Tom works at ExxonMobil. He's an environmental scientist at ExxonMobil Biomedical Sciences, the same organization that Roger worked for before he retired. This organization serves a corporate center of excellence for human health, environmental sciences, and product stewardship expertise. Embassy, as it's known, has a dedicated environmental lab where environmental fate and effects research is formed to support our products, ExxonMobil's products and operation. Like Roger's presentation, Tom's talk will also draw on decades of lab testing experience. So, Tom, you're on mute right now, but you're bringing up I can take you off mute if you want to work on getting up. There you go.
1 second, let me just stop sharing and share. Okay, is that?
There you go, coming up, you're perfect.
Great, okay. Well, good morning, folks. Thank you very much for giving me the opportunity to share with you a little bit about oil toxicity testing. I have a fairly robust amount of material to get through, so let me just jump right into my first outline. So just to give you a quick overview of my talk, what I'd like to do is provide just a brief description of toxicity testing basics. I'd then like to cover 3 key challenges associated with oil toxicity testing. I'd then like to discuss some guiding principles that we found to be really helpful in understanding oil toxicity testing. And then based on those insights, identify 2 key opportunities that we see can be used to help improve oil toxicity testing. And then provide a quick case study to show how these ideas can be practically applied and integrated into toxicity testing. And then I'll conclude with a brief summary and some recommendations. So a key tenet of environmental risk assessment is that the risk of a substance is a function of both the hazard and the exposure. And the purpose of toxicity tests are really to quantify substance hazard, right? Toxicologists want to kill stuff, so they design experiments accordingly. And there are really 3 basic reasons why one might consider doing a toxicity experiment. I've listed those here. The first is to compare the hazard of different substances for a specific organism endpoint. So for example, is oil A more toxic than oil B to mice and shrimp? A second reason might be to assess the relative sensitivity of different species or life stages or endpoints for a given substance. So are mice and shrimp more sensitive than fathead minnow when exposed to oil A, again, under a well-defined set of laboratory exposures? And a third reason, and I'll pick up on this in my presentation a bit later, is to calibrate and validate models to predict effects under defined lab exposures, so that one can then take those models and make projections about effects under different exposures. And that's particularly important because exposures in lab toxicity tests don't reflect exposures in the field. And if you think about it from a lab toxicity test perspective, the exposures are typically constant. They're conducted in closed systems that reflect equilibrium conditions, and they're often on the time scale of days or even weeks. In contrast, exposures in the field tend to be highly dynamic. They occur in open systems and reflect non-equilibrium conditions, and they often occur on the time scale of hours, sometimes even minutes. And this is illustrated in the plot that Tim presented in his seminar last week, and Roger also referred to it. It's a paper by Adriana Bergiano from 2014, and it shows on the y-axis oil concentration, on the x-axis time after a spill, and you see that the oil concentrations that are detected underneath the slicks decrease very quickly to less than 1 ppm in the matter of within roughly 1 hour. So the key take-home point from this slide is that toxicity tests alone cannot define actual risk. And nevertheless, we see that this point is misunderstood, and it's a common flaw in literature studies that either conclude or imply that the effects that they're seeing in these lab studies must be occurring in the field. So before I jump into oil toxicity data and sort of the methods and some of the challenges, I thought it might be instructive just to quickly spend a moment on hazard data for Corexit 9500, which is one of the most common dispersants that are used. And I have a cartoon on the right that shows sort of the design of a simple toxicity test. So what usually is done is organisms are exposed to increasing concentrations of dispersant, and then that data is used to calculate an effective concentration, or EC50, that causes a 50% response, adverse response to the population that's tested. So in this case, the EC50 would be 10. In fact, it's an LC50 because its lethality is the endpoint that's actually being measured. And if you go to the literature, there are hundreds of toxicity tests that have been conducted for Corexit 95. Arguably, it's one of the most best tested chemicals on the planet. And I've shown provided some references that provide recent reviews. And one can take that toxicity data and construct the probability distribution that's referred to as species sensitivity distribution to essentially calculate safe concentrations, as illustrated in the plot on the right. And you can see from that analysis is that acute effects of dispersants don't occur until you get to concentrations that are above about a part per million in constant exposure tests. And if you were to actually do the declining exposure tests, toxicity would even be lower. So, you know, based on typical field application rates, concentrations of Corexit 9500 in the field will just not be high enough to really cause adverse effects on aquatic life. And there's, the last point is that there's a growing database of hazard information from other oil spill response agents that can also be used to do these sort of calculations. Okay, so let me talk a little bit about oil toxicity testing. And Roger mentioned this. The most common approach for conducting oil toxicity tests involves using water accommodated fractions. And to prepare a WAF, one basically takes a known amount of oil and adds it to a closed test system. One then mixes to allow the hydrocarbon components in the oil to equilibrate with water and the overlying air that's in the headspace. One then stops mixing and allows for a settling period, typically about 6 hours. And then one decants the water from the bottom of the vessel for use in toxicity testing. And so the WAF contains both a soluble phase, which consists of dissolved hydrocarbons, as well as droplet oil. And the amount of droplet oil really depends upon how you do the mixing. So if you see the picture on the left represents just physical mixing, you see there's very little droplet oil in that WAF. Just based on you can just note how clear it is. In contrast, if you use that same physical mixing with a dispersant, you get much more entrained droplet oil, and you can see that appears as a chocolate brown. And there's actually other dosing methods, for example, the high energy WAF protocol, where you actually take the oil and you entrain it in the water using a high speed mixer. So that results in even higher droplet concentrations. So that brings me to some of the key challenges associated with oil toxicity testing. And the first key challenge is just the sheer number of studies that have been published. And this graph just shows the publication trends over the last 3 decades. And you can see that post Deepwater Horizon in 2010, there's a significant increase in the number of oil toxicity related studies. And this presents a real challenge to decision makers, since they have to sort of wade through all this information. And very often a number of these studies will have very divergent conclusions. And so this in itself creates a real challenge. A second important challenge is that different dosing methods are used. And so this slide sort of helps try to illustrate that there are basically 2 methods that are commonly employed. The first method involves variable loading, in which different amounts of oil are equilibrated in a WAF test system, like I showed you in the previous slide, and then those WAFs are then used to expose test organisms. And that approach involves one equilibration. The second approach, referred to as variable dilution, uses essentially a single WAF stock that's generated at a high loading, and then makes dilutions of that stock WAF, and then exposes the test organisms to those dilutions. And it's important to point out that the variable dilution protocol involves 2 equilibrations, right? Because the droplets that are carried over from the stock WAF then become diluted, and now those droplets have to re-equilibrate. So that's a second equilibration. And the key point that I want to highlight here is that the actual exposures that result from these 2 different approaches are different, and they're not comparable. So this chart just tries to help reinforce the importance of the dosing method on influencing WAF composition. So there are 3 WAFs depicted here on this slide. They're all prepared with the same oil and at the same loading of 1 gram per liter. And the first plot shows a low energy WAF that involves physical mixing alone. The second plot shows a chemically enhanced WAF, which is based on physical mixing plus adding a dispersant. And then the third plot represents a high energy WAF where the oil is mixed with a high speed blender. And on the y-axis is the percent composition of 50 individual PAHs that have been measured in each of these WAFs. And on the x-axis is the actual PAH analyte. So the pink bars represent the parent and alkyl naphthalines, the green bars represent the parent and alkylated fluorines, and then phenanthrines, and et cetera, to the higher ring, lower solubility pH homologs. And so what you can see from this analysis, and I've also included the total concentrations, the dissolved concentrations, and then the fraction that's dissolved, which is just essentially the fraction of the filtered versus the unfiltered measurements of the TPH50. So that's the total sum of those 50 pH analytes that have been measured in the WAFs. So what you see is that total concentrations, the dissolved fractions, and the composition of pHs varies widely. So in the Lee WAF, essentially 99% is dissolved. In the He WAF, only 4% is dissolved from a fraction perspective. But if you actually look at the absolute concentrations, the dissolved concentrations are quite similar, right? And that's consistent with Raoult's law. It just demonstrates that equilibrium has been achieved. So although the total concentrations are very different, the dissolved concentrations are quite similar. Again, just showing that at equilibrium, the dissolved phase concentrations are consistent. So these data really highlight the modulating role of droplets on WAF chemistry. And an important implication is that the use of the variable dilution method yields inconsistent dissolved oil exposures as the droplets will dissolve. And then I've tried to illustrate that in the subsequent slide. And what this shows is the PAH percent composition of a He WAF that's diluted in a series. So you have a 10% dilution, a 1% dilution, and a 0.5% dilution. And the first panel represents the 100% stock WAF. And again, same setup as the previous slide, the percent composition of the different 50 PAHs, and the x-axis indicates the individual pH analytes. So what you see from these data is that the total concentrations and composition is independent of dilution. That's the black dot. So the total concentrations, right, they're going to dilute directly with the amount of dilution. However, upon dilution of the stock He WAF, oil droplets then re-equilibrate. And this droplet dissolution effectively buffers the dissolved phase concentrations, so that the fractions of pHs dissolved increases as the dilution increases and the composition changes. So you can see that in the He WAF, right, you start with 1.5% dissolved, but if you dilute 200-fold, more than 50% now is in the dissolved form. So again, a 200-fold dilution of the He WAF only decreases the dissolved pHs by less than 7-fold. And so the take-home message, this is an important concept, is that droplets can significantly complicate the interpretation of variable loading tests, because the dissolved oil is really what drives toxicity, which I'm going to highlight in a subsequent slide. A third key challenge is inconsistent exposure characterization. And when you go to the literature and you look at oil toxicity studies, you'll see lots of different exposure metrics that are reported in interpreting oil toxicity tests. So there's nominal loading, there's percent WAF dilution, there's total petroleum hydrocarbons, and different methods are used. or total petroleum, total polyaromatic hydrocarbons. So those are the PAHs. And in the example I gave you, it was TPH 50, but you'll see other papers where they use a different subset of PAHs. And the key shortcoming of these traditional exposure metrics is that they don't differentiate dissolved from droplet oil. They don't account for a wide variation in the potency of the individual hydrocarbons that really can span several orders of magnitude. BTEX has very different toxicity than chrycine. And a last issue is that these approaches don't quantify a whole host of unresolved hydrocarbons that are part of that UCM that can contribute to toxicity. So the real practical implications of this is that differences in dosing methods, which I talked about in the previous few slides, coupled with this lack of consistency in exposure metrics, really confound interpreting and comparing oil toxicity studies. And as a result, depending on the test design and exposure metric, very different conclusions can be drawn. And I've given, in the backup slides, I've given some examples of how you can see very different conclusions are drawn based on the types of exposure metrics that you interpret your oil toxicity studies with. So that leads me to providing some guiding principles for how one can understand oil toxicity tests. And so the first key principle is that the composition and concentration of dissolved oil dictates the aquatic toxicity. And the reason why is that the dissolved oil is what's bioavailable to the organisms. And because the toxicity of the individual hydrocarbons really varies, the composition really matters. A second key principle is that the dissolved oil exposures in laboratory tests really depend on how you do the experiment. So it depends on the actual test oil that you're using and the composition. It depends on the waft preparation method, and I think I've tried to highlight some of the why that's important in terms of how it affects the dissolved oil exposure. And also another issue is whether or not you use open versus closed test systems for your organisms, because that will then dictate the extent to which VOCs are retained or lost during the actual toxicity test. And a third principle is that more soluble hydrocarbons drive oil effects under short-term exposures. And that's because the organisms take up the more water-soluble components faster than the less soluble components. So in a time variable scenario, the more water-soluble hydrocarbons are more important in driving the observed effects. And so these insights have been incorporated into mechanistic models. One example is Petrotox, which I'll talk a little bit about in the next slide, as well as new analytical tools, namely passive samplers, for improved assessment of oil toxicity. So again, this leads me to my first opportunity, is integrating toxicity modeling into toxicity testing. And I'm going to highlight Petrotox. This is one model that we've been very involved in developing, and I'll just quickly try to walk you through how Petrotox works. One begins with a detailed analysis of the test oil that you're interested in, and then one then takes the mass fractions of those different oil components and maps that to a library of representative structures. That then is used as input into a three-phase WAF model. It's a mass balance model that's based on equilibrium conditions, and it uses Henry's law and Raoult's law as the basis for simulating dissolved oil components in your WAF test system. One then takes those dissolved components and then normalizes those concentrations by the corresponding effect concentrations, the EC50s, which is calculated using the target lipid model. So one then takes those toxic units for each component and then sums them across all components and compares that to a convergence criterion of 1, which corresponds to a 50% effect. And then one reiterates that calculation using different loadings until you converge with one toxic unit. And so that's sort of a very high level overview of how the model works. The target lipid model, I don't have time to go into detail to discuss that. I have some slides in backup that cover that in more detail, but suffice it to say that the target lipid model can be calibrated using a single hydrocarbon toxicity test data. Okay. And a really neat recent application of Petrotox is found in the NASEM, the National Academies of Science and Engineering Medicine Dispersant report from 2019. I've taken this plot from that report. So on the y-axis is the acute toxic units for a sensitive organism, and on the y-axis is total oil concentration. It's actually nominal loading, and the various color graphs represent the calculated toxic units from Petrotox for different fresh and weathered oils. And you see where that solid line intersects those graphs, that corresponds to the predicted concentration where you would expect to see a 50% response. And what should be obvious from this plot is that the predicted toxicity across these various oils really varies by several orders of magnitude. So this really just highlights the importance of composition and how composition really influences oil toxicity. And the last sort of second opportunity is the use of passive samplers as a new analytical tool to quantify dissolved oil exposures. And a really convenient, commercially available passive sampler is a solid phase microextraction fiber. And this is a silicone, it's a glass rod, silicone rod that's coated with polydimethylsiloxane, a thin film. And if you take that fiber and you equilibrate it with a WAF, that coating both extracts and concentrates the dissolved hydrocarbons on that PDMS coating. And then after equilibrium, you can then take that fiber and insert it into a GCFID, and the hydrocarbons, the dissolved hydrocarbons that have partitioned, that have sorbed onto that PDMS film or desorbed onto the column, and then you can quantify that using a flame ionization detector. And so what I've shown here in this plot is these are SPME chromatograms that have been generated by equilibrating fibers in a WAF. This is a pretty light crude oil at different loadings at 640 and 700 milligrams per liter, and that's shown in the different colors. And you can see at the low loading, you really only pick up pretty insoluble components. But as you increase the loading, you dissolve more of those water-soluble components into the water, and you pick that up on the fiber. And then as you go to much higher loadings, right, more and more of those water-soluble components are dissolving. So essentially you're filling, as you increase the loading, you fill the chromatogram up to the left. And so this really is a nice this display of how the fiber is reflecting the multi-component dissolution behavior of a complex oil. And one can also conveniently use the area underneath these GCFID chromatogram curves to quantify the dissolved hydrocarbon exposure. So a very quick case study. So what we did here, this is some work that we did in our lab a number of years ago. We prepared physically and chemically dispersed WAFs with 5 crude oils and number 2 fuel oil. We looked at 2 different dispersants, so we did both WAFs and CWAFs, and this was using a variable loading design. We measured SPME concentrations for each WAF, CWAF loading, and I've sort of just quickly outlined the procedure that we used for measuring the fiber concentrations that reflect the dissolved oil exposures. We also calculated toxic units because we had compositional information on these oils using Petrotox. And in parallel, we determined the 48-hour acute toxicity using mysid shrimp. So here's a quick summary of those results. So let me orient you. The middle panels are the observed 48-hour % mortality in mysid shrimp. And on the x-axis in the first panel is oil loading. The second panel, the x-axis is measured SPME concentration. So this is the amount on the fiber. And then the last panel is mortality plotted against calculated toxic units. So you can see from the first plot, oil loading is really a poor predictor of toxicity. You see that there's a wide variation in those dose responses across the different oils. I should also mention that the different colors represent the different oils, and the open symbols represent chemically dispersed, and the filled symbols represent physically dispersed. So no dispersant. If you look at the second two panels, you can see that both toxic units and SPME really normalize responses across oils, since the dose responses collapse into a nice, well-defined dose-response relationship. And the plot on the right shows that toxic units are correlated with the SPME measurements. This suggests that the polydimethylsiloxane coating provides a surrogate for the target lipid in the organism. And that physically dispersed oil, which is denoted by the filled symbols, really show the same toxicity as chemically dispersed oil, which is denoted by the open symbol. So again, the fact that both these treatments are collapsing into a single dose response relationship really suggests that toxicity depends not on dispersant, but rather on the dissolved oil exposures. Okay, so a quick summary. Toxicity tests characterize hazard, not risk. Dissolved oil concentration and composition really is what drives the aquatic toxicity of oils. And there are exceptions, but by and large, that I think is a pretty well founded hypothesis that is borne out by lots of toxicity data sets. And hopefully I've convinced you that modeled toxic units and passive sampling measurements really provide a path forward for quantifying exposure and better interpreting oil toxicity studies. Lastly, dispersants can influence observed oil toxicity in lab studies. This can be directly if the dispersant concentration goes above 1 part per million, because then organisms, you can actually get direct toxic. If you have a sensitive organism at a part per million, you can begin to see effects just from the dispersant. It can also, dispersants can also influence observed lab toxicity tests indirectly by introducing oil droplets that buffer dissolved oil concentrations. And I think I try to highlight that in the presentation. But if you go back to the beginning of my talk, such lab results are really expected to have really limited real-world applicability, just because the disconnect between the lab exposure and the field exposure. So I'll leave you with 4 specific recommendations. So I would recommend to avoid oil toxicity test designs that are unrealistic. So for example, don't test dispersant plus highly weathered oil or dispersant plus nearshore organisms, because those scenarios don't reflect the use of dispersants. Also discourage using high energy WAF protocols, which introduce an unrealistic amount of droplet oil in the test systems. And this is particularly problematic when you use a variable dilution protocol. A second recommendation is to perform single hydrocarbon toxicity tests for your organism or endpoint of interest. And we recently published some work on this using deep sea species. A big question was, well, what's the relative sensitivity of deep sea species versus shallow water species? And there's not, or pelagic species, and there wasn't a lot of data on that. So we were able to do some single compound experiments on a number of deep sea species and determine their relative species sensitivity. These are much simpler experiments to conduct and interpret. It allows the relative sensitivity of organisms, life stages, endpoints to be easily determined. It also enables the time dependence of toxicity to be nicely characterized. And in of particular importance is it really supports the calibration of quantitative oil toxicity models. A third recommendation is that when you conduct oil toxicity tests, have the goal to use that data to confirm or refine toxicity model predictions. And again, we really would recommend using a variable loading protocol since it's really simpler to model than a variable dilution approach. And this is, I think, also consistent with some of the recommendations in the National Academy of Sciences report. And last, we really would encourage folks to promote use and standardization of passive sampling methods for dissolved oil quantification, since it really is targeting that portion of the oil that's really driving aquatic toxicity. So that's all I have.
Thanks, Tom. We have a lot of questions, but in the interest of time, I want to give Benny his 30 minutes, but I'm willing to stay after Tom, hopefully. Maybe you will, and the other panelists. So if anyone wants to stay after, we'll come back and answer as many of those questions as we can before we run out of energy. But Benny, if you want to start loading up your slides, and I will introduce you. All right, so as I said, Benny's going to talk about some work that he's done to take the kind of toxicity data that Tom discussed, and how do we use that to help with real world decisions, right? And I've always thought that it's tough to give an incident commander during a response a bunch of LC50 data and have him make a decision about whether or not dispersants should be used. But what Benny's going to talk about is a study that he did to determine, with toxicity data measured in the lab, what the population level effects might be on a couple of important fisheries. So he's going to talk about that today. So Dr. Gallaway is president of LGL Ecological Research Associates in Bryan, Texas, and has been a senior fisheries and marine ecologist with the company since 1974. Dr. Gallaway has more than 3 decades of experience dealing with the environmental impacts and effects of nearly every phase of oil and gas development in the Gulf of Mexico. With that, I'll turn it over to you, Benny. And there you go.
I believe I'm unmuted now.
Yes, and your slides are good.
And the slides are clearly visible.
Yes.
Okay, well, this is talk is going to show you what happens when you turn a biologist loose with these data, a biologist that's concerned about fisheries management and maintaining healthy stocks of fish. So statistically unexpected catastrophic discharge events, according to Boehm, like the Deepwater Horizon event, sometimes occur, even though they're unexpected. And although the Deepwater Horizon event spill occurred in deep water, oil from the spill was transported into shelf waters where it impacted valued marine life such as the red snapper, Lujanus capuchinus. And if you're familiar with the Gulf of Mexico, you know that management of this species dominates the issues in the Gulf. The red snapper is one of the most important pinfish harvested from the northwestern or northern Gulf of Mexico. I would say the most important. And as a result of this, its biology and population dynamics are reasonably well known, which enables a reasonable assessment of effects to be made. Now, as I say that, the eggs and larval stages of red snapper, as well as other fish and invertebrates, are the most susceptible life stages to oil spills. The eggs and larvae have restricted motility and cannot readily avoid exposure to oil. In contrast, adult fish can sense and avoid oil in the water column. This is well documented. The Deepwater Horizon oil spill was estimated to have killed some 4.5 billion larval red snapper. These larva losses, when you project these larvae out to adult fish that might have been harvested, translated to a loss of 112,000 kilograms of red snapper production. If you look at the average landings, which are outlined in the second bullet there, you'll find that this loss production translates to only about 2.2% of the typical annual landings for red snapper, not a large amount. However, what the question I asked, well, so what if a large oil spill occurred on the shelf of the northern Gulf of Mexico, particularly on the shelf in an area where red snapper, an area highly important to red snapper? What would be, if it did, what would be the impacts of using dispersants versus letting it naturally weather? In this paper, we try to address these questions using the following approach. First, we defined oil spill scenarios where a large oil spill occurs in prime spawning grounds during the peak of the spawning season for red snapper, worst case. We used an oil spill risk analysis model, and this was the responsibility of one of our co-authors, Dr. Conkle, who determined the distribution and volumes of water having lethal concentrations of oil, which is, you see, is a pretty complex issue from the previous papers. Then within this volume, we estimated the density of egg and larval red snapper within these contaminated waters using the CMAP data. This is Southeast Area Monitoring and Assessment Program, monitoring program conducted by National Marine Fisheries Service. Once we had the level of estimated mortality, we wanted to place these losses into context using the red snapper stock assessment models and other assessment models. So to start with, the Gulf of Mexico, BOEM divides the area into western, central, and eastern areas, and as well as into 5 depth zones, from 0 to 20, 20 to 60, and so forth, as you see by the panel on the right. This area, based on red snapper spawning locations and distribution and settlement of early life stages, the western part of cell C2, which is right here, that was selected as a hypothetical spill site. That's a critical area of habitat. This area would be expected to have extremely high densities of red snappers, eggs, larvae, and early juveniles. The hypothetical oil spill, we decided to involve the release of 225,000 barrels of South Louisiana crude oil over a period of 5 days, 45,000 barrels per day. The hypothetical oil spill occurred on 15 July, which is the approximate peak or median spawning time for red snapper. Placing the oil spill in an important spawning area and timing the spill to occur at the median spawning time set the stage for what we feel is a conservative assessment of the spill impacts. Our oil spill simulations were run for cases with and without the use of chemical dispersants. 2 disbursement scenarios were considered. Dispersal of 100% of the spilled oil and dispersal of 70% of the spilled oil. For the without dispersant case, it was assumed that 4% of the oil was naturally dispersed into the water column. The transport of spilled oil was modeled using the CIMAP, physical fate effect, biological effects model, developed by French McKay. The model describes the physical fates of spilled oil as influenced by processes such as evaporation, spreading, all these things that you guys are much more familiar with than I am. And then ambient environmental conditions were used for predicting the trajectory and concentrations, and these were long-term averages. We used 2 threshold levels. I laugh because I just heard all these discussions of threshold levels for determining the toxic effects of total polycyclic aromatic hydrocarbons, concentrations in the water column. These thresholds reflect concentrations that had been documented to kill 50% of the test larvae when they were exposed for 96 hours. That's the 96-hour LC50. The concentrations we used were the same ones that were used in the assessment of the BP oil spill, and that's 50 micrograms per liter and 1 microgram per liter. And we're going to focus on the 1 microgram per liter. And again, we're using the most conservative of the toxic threshold. And then we took an even more conservative step, and that we assumed that 100% of the eggs and larvae were killed, even if they were exposed to the lethal concentrations for as little as 1 hour. Not 50% in 96 hours. If they saw that concentration in the model, they died. This, we think, is highly conservative and obviously not very likely. based on a long history of use for assessment purposes, the CMAP ichthyoplankton data from 1982 to 2008 were used to estimate the average red snapper egg and larval densities in our impact area over the months June through October. Larval densities were estimated from catch per unit effort data. That's the number per cubic meter of water that were filtered by the nets. And corresponding egg densities were estimated using an egg to larval ratio following a paper we published in 2007. And I note that the same approach was used for the estimation of impact from the Deepwater Horizon oil spill. Same data set, et cetera. About the red snapper, it's a long-lived fish. I don't know if you know that it lives for over 50 years. It's also one of the most fecund fish in the world. Individual females of ages 5, 10, and 15 years are capable of producing 19, 69, and 88 million eggs per season per female. Peak spawning occurs from June to August, as I've indicated. Now, this is important. Eggs are estimated to have a duration of 1 day and a natural mortality rate of about 0.5. That's high. Then we assumed a larval stage duration of 27 days, and that's a published estimate. I think it was also used in the BP assessment, and a natural mortality rate of 0.3 per day. That is also high. The stock recruit relationship for red snapper is characterized by a high steepness parameter. 0.99 is used in the most recent stock assessment. The significance of this steepness level is that recruitment will not change very much except under conditions of a severe reduction in stock size. In other words, a very small stock size will produce the same number of ultimate recruits as a very large stock size, and that has to do with habitat availability for the young fish. And then for basis of assessment, the geometric mean stock size of eggs for the period 82 to 2008, the same year we have data for, was estimated in the recent stock assessment. It's 129,094,606 million. So it's 129 million millions. We're going to report all these data in millions, but you get in such numbers, large numbers, that biologists can't even say them. So we use our unit is millions. Following our 2007 publication, we used the Fecundity Hind casting model to estimate egg and larval losses due to the oil spills, and it's very straightforward calculations, and then our Fecundity Hind casting models projects backwards rather than forwards and converts the total estimates of egg and larval mortalities to equivalent eggs. In other words, we're much more sure of our estimate of how many eggs it took to produce a larvae, so we can get to eggs much easier than we can get to 10 or 12 years old. The equation for doing this is published. It's quite simple. Once you get the calculations completed, the impacts are expressed as a percentage of eggs lost from the total egg stocks and the number of females that would be required to replace the lost eggs. So now let's get into results. The areas and volumes impacted by an assumed effects level, toxic level for at least 1 hour varied by scenario and response action. Here you see the two effects concentrations. Here's the area and the volume. You can see that dispersants affected the area and volume. The volumes were basically an order of magnitude higher for the dispersed oil cases as compared to the not using dispersants. And this is, according to my biological background, simply due to dispersants rapidly transport large quantities of oil into the water column, thereby increasing the volume affected. And now the hypothetical egg and larval equivalent egg losses were calculated as follows. First, we did the larval losses. Remember, these are in millions. Here's the 1 microgram and 50 micrograms. So we're going to concentrate over here. Then you calculate the egg losses. And when you get down to the equivalent eggs, you have 58.826 million million eggs, equivalent eggs that were affected. And if you want to, that's the mean, but if you take the upper 95% confidence interval, it's 75,341 million equivalent eggs. If that exposure to 1 microgram for as little as an hour was indeed lethal, and you lost 75.3 or 341 million eggs, that would represent a loss of 0.06% of the total egg stock. And those egg loss could be replaced by a total of about 14,000 age 5 females, 5,000 age 10, or 3,000. And if you look at how many of there are in the stock, based on the stock assessment, there's 1.2 million age 5, 445,000 age 10, and 33,000 age 15. So the total egg loss could be replaced by a relatively small number of females, ranging from 1 to 9% of the total stock. We conclude that a large spill, as I've defined, would not significantly impact the stock. Further, given the steepness of the stock recruit relationship, the egg stock reductions were far less than the level that would be necessary to reduce recruitment. In fact, you need about an 84% reduction to get a decrease. And our assessment agrees with the Deepwater Horizon assessment that it did not have, that spill did not have a significant impact on red snapper recruitment. That those findings are confirmed by a couple of other published studies, at least. And if you look at some of the data, if we look at recruitment over the period 2000 to 2016, it goes up and down, but it's been the 15-year average. It's relatively constant. If you look at, well, what has the biomass done, particularly from 2010, it's increased. You see it flattening off here. This is because of restriction and catch limits. The population is healthy and growing, and catches are being increased. As you see here, these are the metric tons caught. These decreases are due to regulations decreasing the effort. Again, the red snapper fishery appears healthy even after the stock. The information I presented here that has been presented, has been published. You can find it here. So acknowledgements, literature cited, all of this will be available to you, and I'm open to questions.
Thanks, Benny. That was good timing. We have time for a few questions for Benny. So if anybody has a question for Benny, I'm looking through the list. Let's see. All right, here's one, Benny. Based on your research, have you evaluated or considered how the fisheries closures during the year or so after the spill impacted female stocks? I'm assuming that's Deepwater Horizon.
Read that again, please.
Sorry. So this is referring to the Deepwater Horizon spill, I believe.
Okay.
It says, based on your research, have you evaluated or considered how the fisheries closures during the year or so after the spill impacted female stocks?
Well, it reduced the take, which at that point in time served to contribute to stock rebuilding, in my opinion.
And that's kind of based on the data you just showed as well, right?
That's correct.
Okay. All right, if there's more questions for Benny, because we have time, but while we wait for some of them to get typed in, I'll go back and ask some of the questions that we didn't have time to, or we missed before. But so let me go back and I'll read out who these are to. All right, here's a question for Roger. Are you aware of evidence? So Roger, you need to unmute. Are you aware of evidence or investigations of whether dispersants change the surface properties of oil, that is the interfacial tension, in such a way that organisms have greater or lesser ability to attach to the droplets and degrade them?
I think that's a very interesting question. And I would say it boils back down to the question of, do the dispersants stay with dispersed oil droplets? In the picture people draw is a complete coating of surfactants over the droplets. Frankly, I don't believe that's the phenomenon, but I mean, I have no, that's a religious belief. I think it's pretty clear that anionic surfactants leave the droplets. The DOS incorrects it, leaves the droplets quite promptly. There are photographs of it streaming away in pinned droplet microscopy. But I don't know that there's been good analytical data on that. It's an interesting question as to how long the spans and tweens stay with the droplets. I would add that, just as an aside, that one of those is used as a, or at least a very closely related one in the series, is used as a diagnostic. You know, a large number of organisms can degrade those things and a few can't. It hasn't been sorted out thoroughly for the oil degraders. So I think the simple answer is no, it hasn't. And I think the first question to ask would be, do the surfactants stay with the oil, or do they have a distribution into the water? And again, they're much more soluble than hydrocarbons, but clearly they're soluble enough in hydrocarbons that they act as surfactants. So I don't know that we know the answer at all, but I would begin to address it by asking, how long do surfactants stay with the oil?
But I think you do have data suggesting that whether or not you use dispersants, you get roughly the same biodegradation rate, right? As long as the...
Yes, that's certainly true.
That's right. Okay, here's a question. I think this was typed in when Tom was talking, but what is your opinion on the fact that core exit is not very widely approved for use in Europe?
I think that's for you, Tim.
I can answer it if you want me to. I'd suggest that, you know, I think maybe that question comes from kind of the controversy after the Deepwater Horizon with core exit listing and approvals in the United Kingdom. And since then, that their rules have changed. And they allow core exit 9500 to be listed as an offshore dispersant, but not a nearshore dispersant. In the UK, use of dispersants is kind of significantly more liberal than in the United States. In fact, they have rules allowing it to be used in nearshore environments. In 2010, the listing, the regulations required listing for both nearshore and offshore, and they've changed that to allow nearshore listing and offshore listing. Core exit 9500 had a problem with the nearshore listing because of this limpet that was used in the toxicity tests, and it didn't like being impacted with being sprayed by core exit 9500. But for the offshore test, that limpet test isn't part of the rule, and it is listed offshore. And I believe France and Norway, the other major kind of maritime countries, allow core exit 9500. I would suggest, though, that one reason it's not listed is because of the effort for listing in certain places, right? The dispersants business is not a very good business, right? It's peaks and valleys, and the valleys can be quite deep and quite long. So it's not a very good commercial product. You can't really estimate what sales are going to be from year to year. And in the UK, there's two kind of dispersants that have been broadly stockpiled, and that's one's made by Total, Finisol, OSR 52, and the other is made by a small company called Dasic, Silicon NS. And so those are stockpiled in Europe, and they're approved for use. So it's tough for a company like Nalco in the United States to, and it costs a lot of money to get listing. So I think part of the reason, if they're not listed really broadly in the UK, is because of just it's not the cost reward for doing that is not there, because it, you know, they might have a challenge making sales into a market that already has stockpiles of other dispersants. Here's another question for you, Tom. For variable dilution, is it possible to reliably remove droplets from the initial CWAF so that there is only one equilibrium?
Yeah, so I guess the operative part of that question is reliably. Yes, you can. There are approaches, right, where you can remove the droplets. I know Syntef has done work where they've developed sort of a filtration system. I think they use glass wool or something like that. The concern, right, is that some of these dissolved oil components, particularly the higher pHs, have very high partition coefficients. So it's possible, right, that you could lose some of those during the filtration process. But again, depends on what your objective is. If your objective is to calibrate a model and you quantify whatever the exposure is, then you know, that approach would definitely reduce the problem that droplets you know, introduce by modulating the dissolved oil exposure. So it could be, depending upon what your objectives are, could be potentially a useful strategy.
Okay, great. Hey, we're almost at our time commitment here, so let me just thank everybody, because some people have to leave. Again, as I said, I think we'll try to stay over maybe for 10 or 15 minutes and try to go through some more of these questions for anybody that has the energy to do that. But I want to thank our speakers. This week, I think we had 3 of 3 excellent talks. So I appreciate that, and I appreciate everybody staying on time. We have a lot of questions that I'll try to go through some of them, but I wanted to mention that we are having a webinar next week. I haven't confirmed all the speakers yet, so if you have the agenda in your meeting announcement, I hope that they're all going to speak. But next week is focused more on health-related effects from dispersant use and oil spills in general, and we're going to have 3 talks that have some focus on that. So stay tuned and tune in next Tuesday at the same time. And again, I will send out the meeting announcement to everyone who's gotten a meeting announcement before. And I'm not sure I said this last week, but I just learned that our capability for Zoom is 1,000 participants, and we're nowhere near 1,000. So each week I get more and more people that didn't know about the webinar. So I would encourage people to, if they want to send out the meeting announcement to others, but if they could send an RSVP to me, to my email address, so I can keep track of who to continue to send the meeting invite to, because I'm going to send out meeting invites every week for the next week's meeting. So with that, I'll start going back through some of these questions again, and you know, if you have to go, you have to go. Benny, Roger, and Tom, I shake your head. I'm assuming you're okay with a few more minutes.
Yep.
Let's see. Okay, here's one. Does Tom have any recommendations on how to best interpret, in parentheses, toxicity data to compare lab to field, interspecies, and life stage expectations for trade-off decision making?
Yeah, so the way I would answer that is, I think a general strategy for doing this is to apply a time variable target lipid model kind of effect modeling framework and using toxic units as the right exposure metric in that model framework to compute what effects you expect are in time and space, and then take that output and couple it to the kind of population model that Benny just described. And so if in Benny's talk, right, he just used TPH 50 as sort of a very conservative screening, but I think really sort of a next generation impact model would rely rather on total concentrations, really rely on toxic units. So that would be my recommendation.
Yeah, I just suggest that, I mean, there's been a lot of toxicity testing, as Tom showed, and maybe more toxicity testing may or may not be beneficial. When I started thinking about this, right, if you're making a decision about whether or not to use one response tool or another, if you're the decision maker, being handed a folder or a table full of LC50s may not help you with that decision very much, right? So that's why I had Benny talk, but we've also done, and so Benny's got a good kind of screening tool for thus decision making, right? It's not a very complex tool, but we've also done some work through API with this comparative risk assessment, and Debbie French-McKay has several publications, which goes into kind of greater detail and a broader range of species that can be impacted. But it's fundamentally the same ideas that Tom and Benny have been talking about. But if you give a incident commander information on how the whole ecosystem is going to be impacted, or specific fish species that you're interested in, it's kind of easier, in my opinion, to make a decision if you know that here's going to be the impact with dispersants and here's going to be the impact on the whole population, right? That's the goal, is not to destroy populations or not to have a significant impact, or to have less impact with one response option than another response option. And that's why this kind of modeling work, and that's the reason I wanted Benny to describe this, his work. And I think we have Debbie on coming up in, I'm not sure when, a couple of weeks. Okay, let's see. All right, here's a question. Couldn't the SPME hit a droplet and lead to erroneous results?
Yeah, so that's a good question. So at least in our experience, we really don't have problems with fouling of the fibers when we're analyzing wafts and sea wafts. However, in undiluted he wafts, we can actually sometimes see fouling, and the reason why is the droplet concentrations are so high. But it's very apparent when that happens, because you can look at the chronograms on the SPME and essentially it looks just like parent oil, and your reproducibility sort of goes south really quickly. So when you do have fouling, it's typically at very, very high oil droplet concentrations, and it's pretty obvious that you're having fouling. So it can be an issue, but at least in our experience, it's generally not much of an issue, at least for wafts and sea wafts.
Okay, here's another one for you, Tom. Why not use the SPME mechanism as a means of accurate exposure for toxicity testing?
So I think what John is getting at here is, can you use passive sampling in reverse? And so essentially use passive samplers for dosing, so passive dosing of oil. And yes, it's a very clever way to dose oil in toxicity tests so that you deliver only dissolved oil and you obviate the complicating role of droplets. And you can either use silicone tubing, and you can pump the oil in silicone tubing and then let the oil diffuse through the silicone tubing, or you can use silicone O-rings. You basically soak the oil in O-rings, and then you can use those O-rings to passively dose dissolved oil. We've published, we as well as several other groups have published papers on that, so I'd be glad to share those with you. I think hopefully I understood that question correctly.
Okay. Let's see. How important is the oil composition, droplet versus dissolve, to your investigation? And I'm not sure who that's specific to, but if one of you guys want to, Tom or Roger, want to chime in on that?
I think it depends, right? Both are important. Obviously, the more droplets you have, and again, depends on the approach that you're using, right? If you're using a variable loading approach, then you're not diluting those droplets, and so it becomes less of an issue. But again, oil composition also is important. So that's why you sort of need a model to be able to discriminate how important composition is versus droplets. Because the oil composition dictates the composition of the dissolved oil, and the dissolved oil, the composition is very important, as well as the fact that it's in the dissolve phase. So bioavailability and composition are both important. And you need a model to really answer that question.
Roger, what would you say about that? Tom and I have been talking about kind of equilibrium versus non-equilibrium as it might impact toxicity tests. But how about on the biodegradation side? Is equilibrium that is achieved in a beaker, do you think that's a significant issue, biasing biodegradation studies compared to non-equilibrium for the dissolved phase components, or in the open ocean?
Well, I have to say I really don't know, but my feeling is that there are so many different microbes that, as far as the microbiology is concerned, some clearly prefer to be on surfaces, some clearly prefer to be pelagic. Back in the 70s, there's a lot of work done with surfactants and oil plumes underground, and different surfactants helped some organisms and at the same time inhibited others, and that changed. And that was thought to be to do with that the different organisms had different preferences, and also the competition with whatever surface active molecules the organisms were making, in my opinion, stuck to themselves, but potentially also free in the water. As far as the degradation, as you pointed out, we see no significant difference in the degradation of any of the molecules we routinely analyze, with or without the dispersants being there at the levels we talk about. And we've done experiments with massively higher surfactant levels, and it hasn't seemed to affect anything much at all. So the organisms that are degrading the oil, whatever they are, they don't seem to be too hijacked by the presence of tasty other surfactants added. but it could be different organisms, right? I mean, there are so many different organisms in there. Who knows what's doing what? And I think the focus for us is to have the overall phenomenon so that we can say what the phenomenon is, not exactly name, rank, and serial number of the organism that's doing it.
Okay. All right, here's another question for Tom. And I think there's two more that I'm going to go back to, but so I think that might get us through all of them actually. But Tom, what about the unmeasured or unknown oxidized PAHs?
Yeah, so that's a good question. There's a lot of R&D right now on sort of the fate and the effects of the oxidized hydrocarbons, both from the biodegradation metabolites as well as photooxidation products. And so what happens to those? How do they contribute to toxicity? It's a challenge to model all that, right? Because you have all these different, you have tens of thousands of compounds that can all essentially go into different directions. So it's tough to model, but one real practical way to tackle that is to integrate passive sampling in degradation studies, either photooxidation studies or biodegradation studies, because the fiber will pick up at least the nonpolar metabolites that are formed, and then essentially that contributes to your dissolved FID signals. So you're essentially integrating the contribution of those metabolites that are formed over time. And we found a really neat trick is a lot of the photo products are actually organic acids. And so a neat trick is that you can also pH adjust your sample over time and protonate those organic acids so that they partition onto the fiber, so that you can not only capture the nonpolar photooxidized products, but also the acidic metabolites and capture that on a fiber. So again, we think that that's probably the most practical approach to really sort of get at the potential contribution of these kinds of metabolites, whether they be either photooxidized products or biodegradation metabolites, to oil toxicity as it weathers over time.
Yeah, I'd add to that. So this whole issue of photooxidation and how important it is, I think, is something that's kind of become more apparent in the last few years since the Deepwater Horizon, and for a lot of fate mechanisms, right? For example, standard emulsification tests or protocols to study the emulsification of oil is done in a lab without exposing the oil to sunlight or simulated natural sunlight. And so the findings from those tests may or may not be representative of the real world. So we are looking through industry and others, looking at how we can do experiments under more representative conditions, just to understand the fundamentals of emulsification and do photooxidized products that form on surface looks in sunlight. And how does that impact emulsification? Does it make emulsions more or less stable? Does it cause the oil to behave differently as far as spreading? That's something that we're looking into right now. But I would suggest that maybe emulsification for these longer term, days to weeks fate effects is something that hasn't been studied as much as it maybe needs to be studied, and it's an area for more work as we go into the future. Let's see.
Tim, can I answer a question that Alison Chu asked about different crude oils?
Yeah, I was going back to that, so go ahead. Let me read it.
I think it's a very interesting question about the different crude oils. As far as the GC measurements go, right, which is what we measure when we're doing these experiments, the GC composition of different oils is remarkably similar. The density of crude oils, in general, the heavier crude oils just have more asphaltenes and polars. So as far as the GC is concerned, they look just the same. And as far as we can tell, we haven't done many really heavy oils, but these, the biodegradation of, say, phenanthrene goes on regardless of whether it's in a light oil or a heavy oil. It doesn't mind whether the phenanthrene's in diesel or whether it's in a really quite heavily evaporated, for example, the very evaporated Alaska North Slope oil that's sitting on shorelines in Prince William Sound. The rates, are quite comparable. So I think as individual components are concerned, As long as they are dispersed as droplets, then you get rapid degradation. If they're not dispersed, you get essentially infinite preservation. I wanted to emphasize that when we cleaned up the Exxon Valdez spill, oil that had been on the shoreline for a year had essentially undergone no biodegradation since it had landed, because it was so nutrient starved and there was so much of it. As soon as we added nutrients, biodegradation took off very rapidly. So it's really, in that case, the biodegradation was inhibited by the absence of nutrients. In other cases, you can imagine it's inhibited by it's such a thick layer of oil that the phenanthrene in the middle of the oil has no idea there's a Pacific Ocean out there. And I think that question of how fast molecules move within oil, perhaps the surface of a surface oil, a big slick of oil, perhaps the surface is rapidly undergoing biodegradation, but the next phenanthrene takes months to get back to the surface. So the effect is you don't see much degradation. We really don't have an idea of that mobility of molecules within the oil.
All right, unless I'm missing something, I think we have one question that we didn't get asked, so I'll ask it and maybe answer it unless you guys want to chime in. But so are there any opportunities to study dispersant in a natural environment under natural conditions? There are very, very limited opportunities. Next year, there's going to be some, hopefully, they can get permits off the east coast of Canada to do some experiments through the Multi-Partner Research Initiative. You can go online and look that up. But they are going to use dispersants and test a number of different scenarios for how dispersants are used. The challenge with studying fate and effects of dispersed oil offshore is extreme, right? You know, these biodegradation experiments in the lab can last 60 days, and there's just no way to keep instruments in a dispersed oil plume that's traveling offshore for even more than a day or so. All right, it's very challenging, right? Finding the plumes and so instrumenting and taking measurements in the field for the kinds of things we're talking about, even the toxicity studies, which are generally shorter, but just keeping instruments out in a very chaotic environment like the open ocean makes it extremely challenging to do anything but the most rudimentary analysis of what can be going on. So during field studies, I would suggest that probably during Deepwater Horizon, that's, you know, there were studies that went on for weeks and weeks and weeks, and samples were collected many, many kilometers away from the oil spill, from the release point. And so there is information from some of the studies that came out of that, the direct measurements, but it's tough, you know, you can't plan an emergency like that to do experiments. And the next incident will probably be nothing similar. The next significant incident will be nothing that's even remotely similar to the Deepwater Horizon. But I do understand that the researchers have taken lessons learned from Deepwater Horizon. So I think the science that happens on the next major spill, if it's in an environment like the Gulf of Mexico, will be faster and maybe more robust. Did you guys see any questions that I missed? I think I actually got through all of them, which is fantastic. Yep, I don't see any. So we asked all of these. So, okay. Well, there were a number of people that stayed for the questions even, so I'm happy with that. And we had good attendance, almost, 180 people at the peak, it looked like. So this is working out pretty well. I appreciate everybody's attention, and I want to thank the speakers, and we will convene again next Tuesday at 9 a.m. Houston time. So I'm going to sign off.
Thanks, everybody.
Bye.